All right. Happy Friday, everybody. Welcome back to our coregonine science webinar. And before we get introduced today's speaker, Nick Boucher, our former host, is going to take the stand and advertise another webinar opportunity.
Thanks, Bill. In honor of October and the beginning of spooky season, I'm reaching out to you from beyond the grave. Just to let you all know about a webinar that the Quantitative Fisheries Center at MSU hosts about every other week. It's sort of irregular. But the goal of these webinars is to present presentations that are quantitative in nature and related to fisheries in some ways. So the previous two speakers that we've had are Michael Runge, who presented on the expected value of perfect information, and Marie J. Mathey, who presented on some hidden Markov model work for analyzing movement data. And so these are, for the most part, open to the public. We have a few lab-specific trainings that are not open to the public, but I just wanted to let you all know that you're more than welcome to join. If you'd like to be added to the distribution list, I put my MSU email in the chat. And our next presentation will be on Halloween at 1 p.m. Eastern. And Anna Poulton, who is a PhD candidate at Cornell University, will be presenting on some time-varied mortality estimation, as well as methods for optimizing dynamic spatial closures of marine sanctuaries. So if you want to be added, feel free to shoot me an email and I'll get you on the list. Thanks.
Thank you, Nick. All right, speaking of webinars, we need to start filling out ours going forward. I have a lot of ideas of who to reach out to, just returning from the Coregonine Symposium. And so you may be getting an email from me shortly if you gave a talk there. But again, also, reach out to Renee or myself if you would like to volunteer yourself. This is just a great venue to share your results, unpublished or recently published. We're really open to anything, and we like this. We hope that you will find it to be a good venue to get some feedback and hopefully a polite and friendly setting among your peers. And with that, I'm really, really excited to introduce today Nikki Berry. And so I'm actually have the honor of being on Nikki's PhD dissertation committee and have gotten to see her work firsthand. And I think you're going to be really interested to see what she shares today. Nikki got her Bachelor's of Science degree from Ohio Northern University back in 2016, and then went to Miami University in Oxford, Ohio, and where she completed her master's. Her master's thesis was on mosquito larvae and to what extent dissolved organic matter can protect mosquito larvae from ultraviolet radiation. And then for her PhD, she stayed in Craig Williamson's lab at Miami University and has pivoted to looking at the effects of ultraviolet radiation on native and non-native fishes. And so today, you can see there by her slide, exposure to UV radiation induces hatching and alters growth and development of coregonine eggs. And I think her talk is around 40 or 45 minutes, and Nikki's agreed to stay on afterwards for what we hope is an interesting discussion, a question and answer session. So Nikki, welcome, and look forward to hearing your talk.
Thanks, Bo. It was a great introduction. So as Bo said, I'm going to talk to everyone today about the effects of ultraviolet radiation, or UV, which is how I'll kind of reference it for the rest of this talk, on inducing hatching, altering growth. I actually may not actually get to talking about it today, but also the development of coregonine eggs. And I'd like to thank all of our funding sources that I have listed here. Outside of Miami University, we have funding from the Midwest Climate Adaptation Science Center, the Great Lakes Fishery Trust, and the U.S. Fish and Wildlife Service Great Lakes Fish and Wildlife Restoration Act. I also like to thank all of our collaborators, many of which I think are on this call, and our partnering agencies and organizations who have helped us throughout this project. A lot of what I'm talking about today are probably kind of snapshots, bits and pieces of my dissertation chapters, and from which a bunch of these people have helped to gain these data. I also like to take a moment and recognize the Sault Ste. Marie tribe of Chippewa Indians and the Lower Traverse Bay bands of Odawa Indians' right to fish and carry on their ancient relationship with Odonapi and Atikamekw. If you're not really sure what I mean by this statement or what I'm saying, this talk's recorded. Look at the QR code and try to look up these websites, the website on that QR code. But these are two of our tribal partners, and this is something that we've kind of looked into a little bit more as part of my dissertation. I also want to draw attention to an infographic that we recently developed with the collaboration of through the Midwest Climate Adaptation Science Centers with the USGS and Miami University. We worked together and developed this infographic. It's really good. I'll bring it up at the end, at least the QR code for it, so you can look at it in further detail. But today I'm really going to focus on kind of one aspect of this infographic, which is really the effect of UV on the eggs and the egg life stage. So many of us probably know or are aware, especially if you've heard any of my talks, but the Great Lakes have increased in water transparency, especially the offshore portions of Lake Michigan, Lake Huron, since the invasion and spread of the Dresenin mussels. These increases in water transparency could be increasing multiple wavelengths of sunlight. So far, much of the literature has really focused on the longer beneficial wavelengths of sunlight, like photosynthetically active radiation or visible light. But not a lot of data exists and or papers have really looked into the long-term changes in offshore UV and or just collecting UV data in general on the Great Lakes. And just to kind of really drive this point home, this table that I have pictured here on the left, these are essentially the only 4 sites that we have come across in our searches for UV data, where in offshore habitats of the lakes, there's been some type of resampling event in more recent years. And of these 4 sites, 2 of them have had increases in UV and 1% PAR depths, but you kind of have to take this with a grain of salt because it's literally only 2 data points. It's not great if you're a long-term person or looking for long-term trends. So we kind of pivoted and thought, okay, if we don't know how the lakes have changed necessarily in UV exposure over the years, what is the risk of UV exposure in the Great Lakes in the current day? So with the help of all of our partners, again, many of you are probably on this call, we started to characterize the UV exposure in the Great Lakes. And this map here is kind of a map of all the sites that we've sampled by the different organizations that helped us sample those sites. And I'll just put in a plug here that, if you do have a site or you have a location or I convince you by the end of my talk that UV is important and you want to measure it, shoot me an email and or get in contact with our lab and we'll be happy to try to get more UV data. Some of the sampling efforts that we've done to collect UV data have been part of a larger objective. So an example of this would be in 2021 on the CSMI efforts on Lake Michigan. We measured UV depth, 1% UV depths from the near shore to the offshore across multiple transects of the lake. And in general, we found that UV increases from near shore to offshore. So those offshore habitats tend to be much more transparent than the near shore habitats of Lake Michigan. So we have studies like this. And then we also have just opportunistic sampling events where we've gone in the lakes and we've measured UV. And so in the last, I think 3 or 4 years here, we have the recent data since 2020. All of these data points here on this map correspond with a UV a measurement of UV. And it's color coded, so darker colors are more transparent sites, lighter colors are less transparent sites. And I just kind of want to draw attention to this area here up in northern Lake Huron, where we've sampled multiple sites now, and UV is actually reaching near the bottom, if not reaching the bottom of those of the more shallow spawning shoals, kind of up in that region. And this is also a region where we know Cisco are spawning, so it's kind of the relevance there. So I've talked a lot about water transparency and wanting to know more about what the UV exposure is within the Great Lakes, but I may not have convinced you that we need to know more. And so one of the reasons we need to know more about UV is because it's damaging. It can cause tissue damage like sunburn, which you can see in the hammerhead sharks or even on my feet after a long day in the field. But it can also cause tissue damage and program cell death on developing embryos and larvae. And the fish that are pictured on the right here in green, these 4 different levels are different categorizations of tissue damage that we have recently started staining for as an attempt to quantify more sublethal effects. Probably fish from levels 1, 2, and 3 that have that little of damage are going to survive. They may even be able to repair some form of that damage, but certainly level 4, where the entire body is that bright green color indicating a lot of damage, and you actually have parts of the tissue beginning to slough off of the fish, that fish is not going to survive. And that brings us to these more extreme cases where UV can be so lethal that we're seeing actual body deformities where the tails and the vertebrae start to twist up on each other, on itself, and kind of form what I call this corkscrew body form. And on the left, we have alewife, which I know aren't necessarily the focus of this talk, but they also, for the experiments we've done, they also have had an parasite that can colonize them and is not killed by UV. And so there's potential there for some other interactions with the environment in UV that could cause even more harm than good. We know from other work I've done for my dissertation, we've quantified the UV tolerance of multiple species, populations, and life stages of fish. So I'm going to walk through some of those results here. For these figures, the x-axis is either the species or the population of fish that was being tested. The y-axis is what we call the UV tolerance, which is really just the lethal exposure level at which 50% survivorship would be predicted. And that dashed gray line going across horizontally across the figure, that corresponds to approximately a day's worth of UV exposure damage to a Daphnia in the surface waters of a lake, which, without getting into too many details, that can be used as an approximate estimate for a day's worth exposure of UV for larval fish as well. Our take-home messages when comparing amongst UV tolerances among species is that coregonine eggs have a high UV tolerance, and both native lake whitefish and invasive alewife larvae have very low UV tolerances. And if you're familiar with the dynamics in Lake Michigan right now, lake whitefish and alewife both have been decreasing as far as spawning or recruitment of populations from the main basin portions of the lake. So this may be, one underlying reason causing those declines. Looking at UV tolerances among Cisco populations, we tested 5 populations of Cisco, 3 from the Great Lakes, being Lake Michigan, Lake Huron, and Lake Ontario, and 2 from inland lakes. Crooked Lake is in Indiana. It's the southernmost population of Cisco. And Lake Coronis is in Minnesota, and that's actually a Tulaby form of Cisco. When I say Cisco for this talk, just know I'm talking about Coregonus ardii. I realized I probably should have clarified that a little bit earlier, but that's what I'm talking about when I say Cisco. So all eggs have a high UV tolerance, and there's some variability, but no differences between the UV tolerances of these eggs among these 5 populations. And all larvae the larvae from Lakes Huron and Michigan populations have the lowest UV tolerances. This was pretty interesting to us considering the habitats where these eggs were caught from and stripped from the spawning adults are also some of the more transparent habitats that we've measured of these 5 lakes. So it's interesting that they have the lowest UV tolerance at that larval stage. And finally, we looked at the life stages of the fish, and I color-coded this figure to really drive it home. But the salmon-colored data points, these are all the egg life stages that we were able to quantify UV tolerance for. And then the blue coloration, that is the larval life stage that we quantified a UV tolerance for. And in general, our eggs are almost always more tolerant, than the larval life stage. The one exception is alewife, where it is actually more tolerant at the egg stage, but this difference in UV tolerance between the egg and larval stage is very small compared to the difference in the coregonine life stages. So you might be sitting here thinking, okay, so you're going on about UV, it's damaging to the larvae, it can do all this stuff, but the eggs have such a high UV tolerance. And you'll note that for coregonines, this high UV tolerance is generally higher than a day's worth of exposure to UV in the lake. And that's during summer solstice, which these eggs are also going to be generally spawned in the fall or early winter and incubate under ice. So does UV even pose a threat to the egg life stage? Well, the lakes aren't just increasing in transparency, but the Midwestern region of North America is also experiencing a loss of winter, which could increase the risk of exposure to UV in the absence of ice cover. We also know that some coregonines are known to hatch synchronously with ice out on a lake, and the year class strength can be strongly correlated with years of strong ice cover. There's quite a few theories for why, or mechanisms why this might happen. But we're kind of interested in the potential for ice cover to actually be blocking UV exposure to the eggs. And so if we remove ice cover during that incubation period, do you have UV penetrating through the water column now, and the eggs are getting exposed to UV and potentially having issues with their development and hatching success. So our general approach here was we had 2 populations of eggs that we used for our experiments. The first was from the Little Traverse Bay bands of Odawa Indians. This is our lake whitefish eggs from Lake Michigan and near Cross Village. And on the left panel here, I have circled the UV tolerance of those eggs. And then our second population is our Cisco eggs from Lake Coronis with the help of the Minnesota DNR. And that population's UV tolerance is circled on the right here. We got the eggs, incubated them in the lab. For the most part, they're kept in the dark throughout the entire incubations. Unless we were taking them out for a 12-hour exposure, this is what we call the UV lamp phototron. And we have damaging UV shining from the top down onto the dishes, and then we have beneficial light coming from the bottom, shining up through the bottom of the dishes. We can manipulate the amount of UV or the level of UV radiation that the fish get, and we can manipulate which wavelengths are present, being those beneficial wavelengths or damaging UV wavelengths. And this entire apparatus is housed within an environmental chamber where we can also control the temperature during these exposures. We take the eggs off the wheel, place them into their individual dishes, and then we monitor them daily for hatching, survivorship. And in the case of some experiments, we also looked at survivorship or other variables such as heart rate or pigmentation patterns. And we do have some experiments where we've looked at the yolk sac volume to total length ratios and things like that, but I'm not going to be presenting those today. So our first question is, does exposure to UV induce hatching? We took those Cisco eggs, we exposed them to multiple levels of UV exposure because we knew that the eggs it did seem like they were starting to hatch, but it was unclear to us if this was a response that was dose dependent. In other words, is there a tipping point where if you have too little UV, a response is not detected? If you have too much UV becomes so damaging that the response also cannot happen because those eggs are dying before they have a chance to hatch. What we found for Cisco eggs on this figure here, our x-axis is our days post-exposure at which the eggs had hatched, each color corresponds to a different wavelength of UV exposure, and our gray line that is on the very bottom here, that is our dark control where no UV exposure was given to the eggs. So if we have UV exposure present, our percent hatch are hatching a lot faster, our eggs are hatching a lot faster, regardless of what the dose is of UV that they received. When we did the same experiment with lake whitefish eggs, we actually we adjusted our gradient of doses that we gave, and we started with the 2 lowest doses that the Cisco got in the previous experiment, and then we did 2 additionally lower doses than that because the Cisco lake whitefish eggs also have a lower UV tolerance than the Cisco eggs. And we did detect a dose, some evidence of a dose-dependent response where our 2 higher UV exposures have a higher or faster hatching rate than our 2 lower UV exposures. Those fast hatching rates are much more similar to that of our dark controls. So it suggests there may be some dose dependency happening here and that there's some kind of tipping point, but I think these data still need to, these experiments in general still need to get explored a little bit more before we can definitively say if these are species specific responses or if this is a kind of a broad scale coregonine thing, and maybe for Cisco we just didn't give them a low enough UV dose to detect a tipping point there. Our next question is, does the age at which eggs are exposed to UV matter? So our short answer here is yes. These are with the lake whitefish eggs. This plot's a little complicated, but I'll walk through it. So the x-axis for this plot is actually the UVB exposure level. Our y-axis is the average age at hatch. So eggs that were hatching earlier are going to also be younger. And our color coding here is the date of the experiment. The darker color is an earlier experiment, so those are younger eggs. And for the most of our darker colors, those younger egg experiments also hatched at a later age. For our later experiments that we had, those are a lighter color purple here. Those eggs were older when they were exposed to UV at similar doses, and they hatched earlier, 10 to 20 days even earlier than when the young eggs were exposed to UV. And in the case for our eggs here, we actually found that our UV tolerance experiments, we have a cutoff date of 5 days after the exposure at which we will assess their LE50 value. This experiment here suggests that if you were to extend out until those eggs had hatched, those early or younger eggs that are exposed to UV don't actually make it to hatching. They don't survive. So they end up dying before they hatch. So that was another thing that we found with this experiment. Okay, we also wanted to know, do the wavelengths matter? If any of you have worked in a hatchery before, you may be familiar with kind of this notion that you can give eggs a kick. And if you give them a kick being an increase in temperature, maybe you take them off if they're rolling, you take them off the oxygen for a few minutes and just kind of stress them just enough that they respond and they're going to respond by hatching. Well, that kick can also include showing them the light. If they're kept in complete darkness for most of their incubation and then you suddenly expose them to light, those eggs might hatch. So we did an experiment where we actually manipulated which wavelengths of light the eggs were being exposed to. We did this with Cisco eggs, and our x-axis here is days post-exposure at hatch. Our y-axis is the percent that hatched. And what we see is that the blue and gray lines, which are our 2 treatments where UV was not present, you have a very slow hatching rate. Whereas our yellow and our purple lines where UV is present, you have a much faster hatching rate. And it does seem as though there's a slight increase in faster hatching rate in the presence of UVB and photo repair, which is that beneficial longer wavelengths of light. So that does have some effect there, but only in the presence of UV. We also wanted to talk about or think about the condition of the larvae at hatch. And condition here, we're really going to define as being the heart rate as well as the pigmentation patterns. mainly because heart rates can be signals of physiological stress. Also, the pigmentation patterns can provide insight to potential issues or tissue damage pathways, since pigmentation, and this is something that's been looked at somewhat extensively with zebrafish, but pigmentation patterns, molecular pathways are related with the UV damage and UV repair pathways. So alterations in pigment can give insight to that. So first we just looked at survivorship and the percent dead, and we found that our 2 UV treatments did have some eggs that actually had died and did not survive to hatching. Or if they hatched, they didn't survive 2 days afterwards to be able to get a heart rate and pigmentation pattern. So they were surviving. They may have hatched, but in very poor condition. Evidence of this, we see that this figure here, the x-axis is days post-exposure at hatch. Our heart rate is our y-axis. We know that our 2 treatments, the blue and the gray treatments that do not have UV present, have eggs that hatched much later, and they also have a much faster heart rate. In our UV treatments, the eggs hatched very early, but of the eggs that hatched later, they actually ended up having a significantly slower heart rate than the heart rate of the eggs that hatched later in the non-UV treatments. And so we took this as potentially, again, signs of dying. What we call this dendritic patterning of pigmentation in the melanocytes on a fish, larval fish for Cisco particularly, we call this dendritic patterning, but we also see other forms of pigmentation patterning on a fish. And we found, we expect that this dendritic patterning is going to be present. That's kind of our baseline. And so if we saw a deviation from this, that may be a sign or indication of potentially some sublethal effect, whether it be just a consequence of age or UV directly. So of our 4 patterns in our UV treatments, all the eggs hatched much earlier, and they had spotted and dendritic patterning as their primary forms of pigmentation patterns. Our other 2 treatments where UV was absent, however, had all 4 types of pigmentation patterns present. And we take these patterns are probably more likely linked to differences in the age at which these eggs are hatching, and differences in the developmental point at which they hatch, more so than a direct effect of UV damage on the eggs or developing embryos. Okay, the last experiment I'm going to talk about today is recall that I mentioned something about declining ice cover, and if you study lakes or are familiar with them, it should be no surprise that when ice is melting off a lake, you might get a sudden increase or slight increase in your water temperature, in addition to now light penetrating through the water column that could have been previously blocked by UV. So there's potential that we have both an increase in temperature that can influence or induce a hatching event, and we have a sudden exposure to UV, which can also induce a hatching event for these fish. And we wanted to know, does one have a greater effect than the other? Is there an interaction? What is the relationship between these 2 environmental variables, that being water temperature and UV exposure? We did the same experiments I've already talked about with the 4 different treatments, where we exposed them to UV radiation or the beneficial light. We have multiple dark controls because we can only do a UV phototron in 1 environmental chamber at a time. These experiments took place over the course of 2 days. The cold experiment took place first, and the warm experiment was on day 2. And on this figure here, the left panel is the cold experiment, the right panel is the warm experiment, and each of the colors correspond to the different treatments. The 3 different dark controls, the dark control dash phototron, that is a dark control that was actually on the UV lamp phototron wheel. The other 2 dark controls were either inside a cold incubator or a warm incubator, where the rest of the eggs were joined those eggs after the 12-hour exposure period. and all were kept in the dark. We did this to try to test the effect of temperature, since we couldn't actually have two environmental chambers going at the same time with different temperatures and doing the UV lamp experiment at the same time. This was kind of our best attempt at doing that. And what we found was that in the presence of UV radiation, there are significantly faster hatching rates for our UV treatments, but the effect of warmer water did not, there's no difference between the cold and the warm hatching rates for the presence of UV only or the UVB and PRR experiments. When we looked at in the absence of UVB radiation, we found that warmer waters did significantly increase the hatching rate. So eggs were hatching faster in warmer waters. But again, this was only true in the absence of UV radiation. So I'll just bring it back just to kind of talked about a lot of different things here, but I'll just kind of try to recap some of the main points from them. So if we think back to this question, does UV even pose a threat to the egg life stage of incubating if these eggs are incubating under ice and have such a high UV tolerance? And I think our answer here is yes. There's definitely potential for UV to be influencing the at least the hatching, if nothing else, of these eggs, the hatching date of these eggs. So just kind of recapping, very little UV data exists long-term across the Great Lakes. We're working on this. Again, if you're interested in getting some UV data, please reach out to us. We're happy to go and collect it. Coregonine eggs generally have a higher UV tolerance than the larval life stages. This was consistently observed in all of our UV tolerance experiments that we did. UV can induce hatching of Cisco and lake whitefish eggs, so this does not seem to be a species-specific response. And in fact, in earlier experiments we did with bloater, we also saw signs of the same response. That's what kind of got us interested in trying to explore and test this UV-induced hatching a little bit more carefully. The dose needed to elicit an effect possibly needs to be at least 34 kilojoules per meter squared. That's about half a day's worth of UV exposure. But again, this needs to be tested more in Cisco because we only notice this dose response in lake whitefish. The age at which an egg is exposed does matter. So older eggs, at least for lake whitefish, hatched earlier. They can make it to hatching at those higher UV exposure levels as well, where younger eggs don't actually survive to hatch. The wavelength matters. UV always induces hatching. Larval condition is different at hatch. Still unclear what exactly that means for these eggs. So I'm interested to see if other people have some ideas or anything, that I would be curious to hear them. And then the last thing that is kind of my favorite result of all of this is that UV increases the hatching rate much more than just an increase in water temperature without exposure to UV. So definitely, I think that argues for at least considering UV during ice out and how that might play a role in the survivorship of these fish. And just to bring this back home to our kind of original conceptual diagram here, that in this infographic that we have, if you take nothing else away from this talk, I hope that you take that UV can impact the timing to hatch, potentially the condition of a larva at hatch. And these impacts happen independent of changes in water temperature. I don't know off the top of my head what exactly this means for the restoration of these fish. I think the fact that these lakes are increasing in water transparency, I don't know if you're necessarily going to stop that or should stop that, but it does seem like maybe drawing more attention to looking at habitats and reproductive habitats of lower transparencies and how those populations may be doing. That might be something close to an answer, I suppose. Okay, and hopefully I didn't go too far over, but with that, I'll take any questions or conversations.
Thanks, Nikki. No, you did not go over it all. And I'm not sure people have a full appreciation for how long you spent on some of these experiments. I mean, you gave us a lot of information I know that you had many sleepless nights as you were working on these and different stocks, and as soon as you got the eggs in. So just a lot of data here we appreciate, and it may take people a little bit of time to soak up everything that you just gave to them. And so if you want to ask questions, you can do that in the chat. You can also just pop your camera on and ask it. And if you raise your hand, I'm probably more likely to see it. And so I guess I'll ask the first, and a lot of what you were doing was focusing on eggs. But one of the things that still surprises me is the difference in larvae exposure, like whitefish larvae being kind of pretty susceptible to UV, and we know they're like positively phototactic, like they go up to the surface. And so maybe just help people understand how that could be true. Why would they want to get up to those surface waters if your experiments talk about their vulnerability to UV, like being more vulnerable than some of the other coregonine larvae you saw? How could they still survive, do you think?
Well, I have two ideas, I guess. So one, I think one thing is if they're swimming up to the surface and it's during the day and it's sunny out, they might not be surviving. That just might be it. They might not be if that's UV exposure is too high. And even in some lower transparency sites, right in that surface layer, you can still get pretty high levels of UV exposure. And so they might not be. That's one possibility. The other possibility is, and I didn't talk about it in this talk, but it's certainly part of the framework for my dissertation and this work is Craig actually proposed this kind of hypothesis called the solar ambush hypothesis, which is essentially that aquatic organisms that cannot need to be in the surface waters or have a positive attraction to visible light in the surface waters are going to swim up to their death in a really transparent habitat unless they can detect and see UV. And not all vertebrates have UV detecting options, so not all of them can actually see those wavelengths and be able to behaviorally respond to them. And I did some experiments, behavior trials with lake whitefish larvae, and did find evidence that they did not seem to be avoiding UV or making any choice in those experiments, where our experiments with Cisco actually from Lake Coronis were making a choice and were consistently picking the lower light intensity or non-UV side of the chambers. And so I do think at the larval stage, there could be some differences in their behavior to UV that might influence their abilities to survive as well. I don't know, does that answer your question?
No, it does. I mean, I've also heard you talk about the ability to sort of be adaptive and do this photo repair. I mean,
Oh yeah.
is that another possibility where I mean, I guess they're still susceptible if you're finding they're dying in your experiments, but could they also like some?
Yeah, if it's like a low enough exposure, I should say we were pretty successful in those experiments with Cisco, but we haven't necessarily been as successful testing for photo repair in lake whitefish just because they have such a low tolerance. It's hard to expose them to a level of UV that isn't just completely overwhelming that repair mechanism. So I think in the lab, we haven't found evidence of photo repair at the larval stage, but I don't necessarily know that that's because, or that's not a result, that means they don't have
No, I
yeah,
follow you. So yeah, this is really,
yeah.
it's potentially really bad news for whitefish to be so susceptible to
Yeah.
UV. So yeah.
Yeah, not a great day to be a whitefish, that's for
Yeah.
sure.
Okay. Well, I see a long name there, Toto P. Williams, 55 Day Williams, with a hand up.
Yeah, that's Todd Williams, and
Hey, Todd.
very interesting, especially since I target whitefish in Saginaw Bay.
Mm.
But I was curious, have you had any thoughts on other species of fish that might be affected, like lake perch or catfish or walleye, or what I mean by that?
Yeah, let me see if I have it here. So our labs tested other species of fish, and I don't know, can you see this slide that I just pulled up?
Yes.
So these are some examples of fish that I think might be of interest to this group, specifically bluegill, pumpkin seed sunfish, and yellow perch. They all have a relatively low UV tolerance. And the UV tolerance of yellow perch, in fact, is actually much more similar, I think, to that of like alewife and lake whitefish. So if that kind of puts some context there. So there have been other species that have been tested within our lab, and my advisor actually just submitted a paper trying to do a broader synthesis of all the different species. But certainly, coregonines aren't the only fish that could be impacted by UV exposure.
What's that one in blue, Nikki, that's so high, so tolerant?
That's rainbow trout. So salmonids do tend to be like rainbow trout, and then also northern pike, which isn't salmonid, but those both have the higher UV tolerances.
And one thing you've sort of noted is that a lot of pigmentation helps potentially maybe with that. Yeah.
Yeah, that's the thought, at least. I haven't been able to quantify that necessarily, but with my experiments, but other people that have looked at pigmentation have found that it does help, or seems to help with protection from UV damage.
Okay, thanks, Todd, for that question.
Yeah, thank you.
Trevor, you want to ask a question, or I just happened to see you up on your camera?
Oh, sure. I didn't have my hand up, but I have tons of questions. I guess the first one, maybe, it's a really fascinating talk. Thank you for this. Maybe the first one is just developmentally, how long do the pigmentation differences persist? Is there any way to see this in later ontogenetic stages if they've been exposed? And just thinking about if you can go into museum collections and see some sign of differences over time.
Yeah, I don't know is my short answer. I actually was thinking about this though. I think from maybe AFS or some conversation recently with folks, but and they were talking about how just IDing fish, the pigmentation patterns were different than what they were supposed to be, or supposed to be for the IDs, and but the genetics are coming back saying, yeah, it's this fish, and they're like, it doesn't look like this fish. So and that's, I think, I can't remember if that was for Cisco or lake whitefish, but that's what made me think of this too, that maybe there is something here that you could see longer term. I don't know that we have the knowledge yet to answer that.
Thanks. Sorry if I missed that somewhere along the way.
Oh, no, you're okay.
So while others think about questions, I really am impressed by the data you had in terms of being able to separate out the warming versus the UV exposure to really nail down that it is that UV exposure. And I mean, I think just to anthropomorphize a little bit, if you're a larvae and you're incubating under the ice, it would make sense that you wouldn't want to hatch until maybe the ice was gone. And so maybe that is the sort of that trigger, that actual sunlight, that UV exposure, that is the one that sort of, that's the adaptation that lets the larvae know it's potentially a safe,
Yeah,
anthropomorphizing. So I think that makes sense. And so the question is, if we're now in a period where there really is an ice cover, I think that does sort of set the stage for an ultimate, what we call match mismatch, right? Where they're going to be hatching sooner, and whether or not the phenology of the plankton and everything else also moves or not could really have implications for their ultimate survival. I mean, and it's just, when you're talking about billions of eggs out there, tenths of a percentage can make a big impact in terms of a strong year class or not. So really cool finding that you were able to show it is UV and not temperature.
and I
have a question for you, Nicole.
yeah, go ahead.
have you thought of, in addition to just ice cover, like ice quality, like now with climate change and the ice is becoming less clear and murkier with the freeze thaw cycles, how that might also affect the larvae and eggs?
So, I haven't looked into this as much. But I do know the quality of ice cover can influence what sunlight, or if sunlight is actually penetrating through ice. So I think if you have thicker, definitely murkier ice, things like that, it's going to be less light and it actually will block it. But if you had like, sheer black ice that's completely transparent, you might still get UV kind of penetrating through. So we say ice generally is blocking it because you also have a thick snowpack layer and all these other things that are happening. So generally it's likely to block it, but there's definitely potential that depending on the quality or what kind of ice it is, you could still have some light getting through. So I think that would be a great like next step to follow up on, is to actually connect. I think all of our experiments, these have kind of been conducted or built in this framework of potentially this loss of ice cover and how that might increase exposure to UV, but it's not necessarily that we have gone out and, tested if the eggs are getting exposed to UV and they hatch earlier and that type of stuff. So I think trying to connect some of these lab experiments with more of the field data would be the ideal, like kind of next step to go for sure. I also thought of something earlier, so when you were talking, I did test this for the egg hatching for alewife, and it gets a little muddier, the results, so I don't have them in this talk, but we did see earlier hatching in the presence of UV for alewife. And so I think that's something that we have a collaborator that works with zebrafish, and they can develop and hatch, just like that, compared to all of these cranings that take months at a time to hatch. And I think that's an opportunity to look at if this is a mechanism, this UV induced hatching is a kind of broad scale mechanism that a lot of fish are responding to, or if this is something that maybe is specific to these cold water species that are actually developing under ice. Because that might give some more insight into if ice cover is actually at all related to this, or if this is just like something that happens. But there are a lot of fish, alewife included, that spawn at night, and certainly in the daytime, you go through a day of developing, and by the next morning, you're getting exposed to light and you're starting to hatch. Is that a cue as well? So it's something that.
Well, that's interesting. Yeah, alewife definitely would not have to worry about ice cover. So yeah.
Yeah.
Trevor, yeah.
Oh, sorry, another hand's up. I'll wait.
Okay, Kaylee.
Hey, maybe I missed it earlier, but just wondering for UVB penetration through the water column in a non-ice scenario. I get that it's very dependent on water clarity and things like that. Did you guys look into the depths that UVB goes in each of the locations that you sampled from?
Yeah, and I don't know, I think I put that table actually in as an extra slide here. Let's see. Well, maybe not. We did for most of the populations that we tested. I guess I don't have that table here. And it doesn't always reach the bottom is like the short answer. So like Lake Coronis, for example, UV only penetrated to about a 1/4 meter deep into the water column. So that's not very deep at all. But the Cisco also can spawn in like really shallow habitats. And then on top of that, so if the eggs aren't necessarily getting exposure to that 1% of UV, when they hatch, what are the larvae doing? Are they going straight up to the surface? Are they kind of staying a little lower? That, is also going to influence then if they're getting exposed to UV or not. So we did take some measurements at the different sites, or very close to the different sites where we got spawning populations of fish, or eggs from spawning populations of fish in each of these lakes. I should have included that table, and I apologize, I don't remember it offhand.
Oh, you're all right. Thank you.
Thanks, Kaylee. And Nikki, maybe just give her a sense of like how deep it could penetrate, like on a Lake Michigan beach versus you know, out in the middle of the lake. How what's the difference there? You had a figure, but
Oh,
maybe
yeah.
people didn't see it as like, just to give them the numbers.
Yeah, so like Lake Michigan near shore to offshore, this figure here, we found, like, for example, down by Muskegon, we found a difference in not even really going too far off into the offshore, just Muskegon Lake and then out into that near shore habitat right there. We found a difference of less than a half meter deep in that near shore Muskegon Lake, and then as deep as closer to 6-7 meters deep in that near shore, right in that near shore habitat. And so certainly along the beach there, if it's a calmer day, not a lot of wave action, so you don't have a lot of turbidity or things like that, you can actually have UV reaching straight to the bottom where the water is only 1-2 meters deep, and lake whitefish larvae are caught in those waters right there. So it's definitely some potential that they're getting fried in the main basin.
And just to reiterate, and do you know how much UV has changed, like from 20 years ago, like before mussels?
No, I mean, the answer is no.
Yeah.
And no. So we have a couple of sites where UV was measured, but that, yeah, they're not, I mean, I don't think you can really confidently draw any trend off of 2 data points, not for, you know, for the entire Great Lakes. So it's kind of one of those things that's a bummer, but I think it also highlights some opportunities that I think, you know, as we get more data, or there's other data that you could try to build some predictive models for UV from like dissolved organic carbon, chlorophyll, turbidity, those type of things, like you would do to build predictions of the 1% par depth in a lake. You could try to do that with UV. And some of those data do exist much, much further back. So we haven't tackled that question.
Yeah. Robin.
Hi, Nikki. Great talk. It was really good. Do you know if there is someone from your lab or really anywhere, if so you talked about like the changes in turbidity, and I'm thinking how different lakes have different reasons for their turbidity. So it's some of those river mouths that might be from more sediment, but over in Lake Erie, that could be from algae. Do you know, is anyone doing or is it out there? Do you know how the UV light changes how far it can reach into the water column based on what's causing the turbidity?
Yeah, that's a great question. And actually, I don't have the data here, but we did find a gold mine of UV data within the nearshore habitats of more the Canadian water, water of 4 of the 5 Great Lakes. And I don't think Todd Howell's on this call, but he's with the Ontario Ministry for the Environment. And he has been sampling UV about once every 4 years in each of the lakes throughout the seasons in these nearshore habitats since 2002 until actually 2022, I think, is our most recent year of data. And I started to look into those, some of the patterns and trends, and we do see that it's very lake specific, dependent on the different water body types. So he has 3 sites. One site that's a main basin lake site, so it's near shore, but it's supposed to be more representative of the actual water transparency of that main basin lake. And then the other 2 sites are embayments, like Georgian Bay would be, you know, near shore sites along there would be embayment sites, or river sites, which could be either sites that are literally in connecting channels and rivers, or between the different lakes, or those are sites that are influenced strongly by those like the river plume inputs of more than likely dissolved organic carbon. And just in looking at those data, I don't have a slide to show, but looking at those data, I have found that like Lake Huron is most consistently the most transparent, whereas, but that's in the lake sites, and the river sites of Lake Huron are even more transparent But then if you go into Lake Erie, it's no surprise, the embayment sites are are really, really low transparency. So it gets more complicated. There's not a clear pattern where you can just say, oh, all rivers are going to constantly be, really low 1% UV depths, shallow 1% UV depths, but they might have very deep 1% pod depths. That happens in some of the lakes, but not all of them. And then it also changes between spring, summer, and fall, which of those kind of sites are deeper in their UV depths. So there's a lot of fluctuation, but I think that's something, what we've done can help us start to pull out some of the patterns, at least if not within a certain lake or even a sub-basin of a lake. We can start to pull out some of these patterns. I think we can, try to make some generalizations about our habitat that we're studying if there isn't UV data available for it. I think.
So generally speaking, then, just to try to make a generalization, if it's more sort of phytoplankton or dissolved organic versus just a sediment based, which one is gonna like stop the UV faster?
So dissolved organic carbon selectively attenuates UV, so it's going to absorb UV much more than the 1%, so much more than the PAR wavelengths of light. But in the Great Lakes, especially in the offshore waters, where DOC concentrations are so low, and then you also have photo bleaching that's happening because of the long retention time, you're actually getting that phytoplankton and chlorophyll or suspended solids, things like that can actually also contribute to the attenuation of UV, more so than in the nearshore habitats. But in the nearshore, it's generally going to be dominated by dissolved organic carbon, just from the terrestrial environments entering into the lake.
Okay. Trevor, you had another question, and then I see Wendy has one in the chat, but why don't you ask your question?
Sorry, I had the same question as Wendy. I was just curious about the spawning reef restoration and choice of substrate, if this should be affected. But
so yeah, one of the things we've been thinking a lot about is if you're talking about a spawning reef that has a lot of cobble, gravel, rocks, whatever in it, and there's all these different little crevices, those eggs could be falling into those crevices and certainly be shaded, just physically shaded from rock cover. And I think that might be another reason why those that type of spawning substrate of spawning reefs are good. I guess, like that could be a reason, in a very simple way, that could be one of the reasons why, versus if you're spawning on just an open beach, kind of, or substrate where it's not really cobbly and rocky, I guess, for lack of better words. Certainly you're having less areas where you could be having some type of physical shading, but you might have more suspended solids and things like that, depending on what the waves are doing and that sort of thing in that habitat and how shallow it is. So definitely, I think that can play a factor. And also, just to build off of Wendy's question too, so there is some evidence in coregonine species, I think European Vendace, I think it was, but some studies in lakes over in Europe, they found that the amount of pigmentation of a fish was opposite, like it was inverse of the transparency of the lakes those larval fish were from. And they looked at this because they thought that maybe pigmentation would increase the more transparent a lake was, as a response to UV, but they found the opposite of that. And so that's really interesting in that it could be actually more of a evolutionary pressure of predator avoidance. So that kind of game of hide and seek. And if you can camouflage with your surrounding environment better, are you better, suited to avoid being detected by a predator? So it may be that there's some curve here or interaction here where at some point you're in a lake that's so transparent, it doesn't matter about your predators because that UV stress response is just so strong that's going to drive how you survive, versus a lake that has such low transparency that, being able to not stand out against that background in the water column and avoid predators is going to be really important. And so at some point there's probably some interaction where one of those two pressures is it's going to switch, right? Depending on the transparency. So that's kind of another thing I think when thinking about the substrate and also just the reproductive habitats themselves, not just the transparency, but what's creating less transparent or shaded habitats might be important in that interaction. Sorry.
It's a cool example. I'm glad you brought up the Vendace one. And then Wendy, did you want to add anything else, or did she address your question? The only thing I think maybe you also were getting at is sort of the rearing environment in artificial rearing, how that can affect maybe pigmentation type, maybe from some work Roger Gordon has done, and maybe you've seen too in our lab. And
Yeah, I think what she said covered most of it. I was just kind of thinking that, as we make choices about, thinking about reintroduction, the tanks all tend to be very clear and shiny. How is that going to, is light bouncing off of that in a way that it wouldn't in the environment that we might need to think about? Is
Yeah,
kind
and
of where I was going. Yeah.
I agree, and also just the type of lights that you have hanging in the lab or the hatcheries, I think can be important. Like a lot of people don't realize, but just your normal standard fluorescent light bulb has some low doses of UV that's actually being emitted, and so that might be good, it might be bad. I don't know. There's some studies that have found other species of fish that are reared under laboratory conditions actually have a lower UV tolerance than those that are reared in a more natural, well, actually their natural habitat, like water body. So there's some evidence that having a more natural condition to grow in is going to better suit you for the actual conditions you're going to experience in the lake. I don't think that's crazy surprising, but UV is just another variable we need to consider when thinking about that.
All right, thanks. And do you know, is that the same for LED lighting too? Because I know that's sort of a popular choice for energy savings now.
Yeah, I'm not as familiar for LED lighting, if that is true or not. I feel like I probably could just look at the, right, the spectral, I don't know, output or something of it, or I'm sure that the manufacturer has some type of paperwork or something, but yeah, I don't know offhand. Sorry.
Great, thanks.
thanks, Wendy. All right, have we hit our saturation point? Are there any final questions for Nikki? All right, well, if not, Nikki, you had nearly 60 people joining. And you're getting some nice comments here in the chat. Thanks so much for making your presentation to us and opening our eyes to this new potential stressor in the Great Lakes, and some of the different responses you've seen across species are kind of surprising, honestly. Seeing whitefish so intolerant. You did also see intolerance with alewife, which we kind of expected, but to see whitefish also not handle it that great, I think is pretty surprising, and something we'll have to think about from a conservation or restoration perspective. So all right, thanks again, everybody, and stay tuned. We will recruit somebody to give a talk next month, I promise. Until then, have a great weekend.